session of this series that occurs every 3 weeks. So again, welcome. My name is Becca Guth-Metzler, and I am one of the co-organizers. I'm one of the co-organizers for this seminar series, along with Albert Fehrenbach, Danielle Simkis, and James Iguchi. And on behalf of the organizers, we'd like to thank the PCE3 co-leads and staff for their support, and also all of you for coming and making this such a fun and engaging event. So just thank you for all the enthusiasm you bring and for being here. So if this is your first time coming, what we do here is we highlight active research that's going on, primarily done by early career researchers. So this is a platform for them to share what they're doing and for us to find out what's going on at the forefront of research. And the goal is also to have a continuing discussion and collaborate and share ideas. So as part of that, there is a discussion portion, and we invite you to continue in the discussion going on by joining our Slack. So a link for Slack will be available later in the chat. And also, if you're watching this on another place or time, you can find our Slack on PCE3's webpage for the seminar series. So the format for today is in 2 parts. The first part, we will hear from, all of our speakers. And in the second part, we move to discussion. So just keep in mind, if you have a question or comment during the portion where our speakers are presenting, to hold on to that thought or write it down so you can share it during this discussion portion. So today we have speakers all from the University of California, San Diego. And our first student speaker of the day will be Josh Arriola, who will speak to us on ribozymes and the RNA world. And also we will be hearing from, as a student, Hannah Rutledge, and about nitrogenase PClusters. But before all that, we'll start off with a bit of a sparking our interest, our scientific interest, with a brief highlight from Dr. Uli Muller. So you can go ahead and share your screen while I introduce you to us all. So Professor Muller studied chemistry at the Ludwig Maximilians University in Munich, Germany. He completed his PhD on the biochemistry of RNA protein interaction in African trypanosomes at the Max Planck Institute for Biochemistry in Munich and at the University of Technology in Darmstadt, Germany. He did his postdoc studies on catalytic RNAs with David Bartel at the Whitehead Institute at MIT in Boston. And since 2006, he has been faculty in the Department of Chemistry and Biochemistry at UC San Diego. His work at UC San Diego is on the selection and evolution of catalytic RNAs, with the 2 goals to study how life originated and to generate catalytic RNAs for therapeutic purposes. So without further ado, take it away, Dr. Muller.
Thank you very much, Becca, and thank you for inviting me to give these introductions. So first, I was asked to give a short introduction on what my lab is doing. And as Becca already said, we work on the general idea of evolving catalytic RNAs to improve their efficiency, or selecting them to make them initially. And the main focus of my lab is on origins of life, but we also improve the efficiency of catalytic RNAs for therapeutic purposes. And let me first tell you on one slide something about our therapeutic RNAs, and then I will tell you a little introduction on origins of life, where you will hear much more about this than from Josh. So this is one slide that describes one of the catalytic RNAs or ribozymes that we designed. They start off from a natural self-splicing ribozymes where the whole ribozyme is just one strand that continues through here. And we found that you can make both this duplex as well as this duplex in trans. Earlier stages were done in this research were done by Bruce Sullinger and others. And we found that if you combine both of these ones, you can generate a ribozyme that we call spliceozyme, because this ribozyme, which is black, can bind to a substrate, which is here in red, blue, and red. It can recognize these splice sites, and then it does what it also does in the natural environment. It kicks out this sequence here and joins these two sequences here. So effectively, what you're doing is you have a ribozyme that can remove internal sequences from a substrate RNA. And theoretically, this could be used, for example, in a therapeutic application where you can repair a messenger RNA that has an internal sequence that, for example, was not recognized by the spliceosome to be removed. And the way we improve these efficiencies, so after redesigning them and testing whether the principle works, then we improve these ribozymes, and we use an evolution procedure where we introduce mutations, we generate a library, transform it into E. coli cells, and then we challenge the ribozymes in the cells to repair an antibiotic resistance gene that allows these cells to grow and then isolate plasmids. And we go multiple times through that route. And here you see one example of one of those evolved ribozymes. This is the parent ribozyme, and this here is the evolved ribozyme. You see the top band here is the pre-mRNA, and the second band here is the mRNA, where the intron was removed. You see that the parent ribozyme generated a lot of side products, and the evolved ribozyme generates much less. So this is one of the examples of how we evolve these ribozymes for improved functions, and we hope that in the long run they can be useful for therapeutic applications. But the main focus of my lab is on in vitro selections of catalytic RNAs of ribozymes to test to what extent could an early stage of life have relied on catalytic RNAs. And the focus since a few years has to do with this molecule here. This is called cyclic trimetaphosphate. And the reason is that this molecule is a prebiotically plausible source of energy that is generated via a number of pathways, including erosion from meteoritic phosphide minerals and other pathways. So we found that this prebiotically plausible energy source can be used by catalytic RNAs to react with their own 5' hydroxyl group, and this can be in very fast kinetics, and you see the primary data here, and this has worked pretty well. And Josh will tell you on this line of projects where we did a number of steps going much further than this, where I think we have some pretty exciting results. And I think with that, I will just leave the stage to Josh, who will tell you some of the exciting research he has done in the lab.
And while Josh is pulling up his screen, I can do a bit of a bio for him. So Josh Arriola is a biochemistry PhD candidate in the Muller lab at UC San Diego. He earned a BS in chemistry from UC Santa Barbara and went to work in biotech for 2 years. He is currently investigating the role that RNA may have played in the early evolution of life, including identifying and characterizing catalytic RNA that could have facilitated the replication of an RNA world organism, and exploring how other molecules may have aided in the activity of catalytic RNA. So take it away, Josh.
All right, thank you for the introductions, Becca and Uli. As Uli mentioned, today I'll be discussing 2 projects that I've worked on in his lab pertaining to this question of the role that catalytic RNA may have played in an early stage of life. So what I think about the research our lab does, I think the one of the major guiding questions we have is, what can RNA do in the context of early life? What are the boundaries of RNA's role in our early life? So the first project I'm going to be discussing is asking specifically, what can short RNA do? So in extant biology, there's a wide range of RNA lengths. For example, you have things like ribosomal RNA, which is over 1,000 nucleotides in length. And then you have things like the minimal hammerhead ribozyme, which is 34 nucleotides in length. But in the context of the origin of life, is there an upper limit of useful RNA length in the period preceding the transition to biology? People like Leslie Orgel and James Ferris have been investigating this question, and what they have found is that under ideal conditions on mineral surfaces, polymerization of nucleotides generates oligos in the range of somewhere between 30 to 40 nucleotides. So the gel on the left, for example, is showing extension up to about 40 nucleotides after 14 days using imidazole as an activating agent. And then on the right, in a follow-up study that James Ferris did, you can see a similar size range of about up to 40 nucleotides in length. And as Uli mentioned earlier, the specific ribozymes our lab is interested in are ribozymes that demonstrate a central role of RNA in the replication of an RNA-world organism. And I think what these two studies are showing is that to investigate the feasibility of this RNA world, we should be focusing on the shortest possible ribozymes. So previously in our lab, a triphosphorylation ribozyme was identified by in vitro selection, as Uli outlined in his intro. Here's specifically how that selection scheme works. So you start with a DNA pool that consists of the T7 RNA polymerase promoter sequence, a hammerhead ribozyme sequence, and then a randomized 150 nucleotide region. You convert this DNA pool into RNA, and the hammerhead ribozyme is able to co-transcriptionally cleave itself off to generate a free 5' hydroxyl. This processed RNA pool is then incubated with triametaphosphate, and any active sequences are able to triphosphorylate their own 5' end. And then you can isolate these active sequences by ligating to a biotinylated oligo and then fishing them out using streptavidin bead pull down. And this cycle can be done over and over again in order to further enrich for active sequences. And so in total in this original study, 8 total rounds were performed, and Janina, who is the grad student that did this experiment, was able to see active sequences being enriched after round 4. Now, one issue with this in vitro selection is that it generates ribozymes in the range of 150 nucleotides, which, as I showed earlier, is not really prebiotically possible according to the literature. So the goal of my project was to identify short catalytic motifs using a combinatorial method. And the way that this method worked was using a randomized primer and the Clinau fragment of DNA polymerase 1 to generate truncated DNA subpoles. So this randomized primer was able to bind somewhere in this region of interest to generate a range of DNA subpoles, anywhere from subpoles that contain the full N150 region to subpoles that didn't contain the randomized region at all. Next, these truncated subpoles were then fractionated by gel purification, and we took 10 fractions and took each of those 10 fractions through one round of selection. The idea being to figure out what was the fraction that contained the smallest possible subpoles that still had activity. And what we ended up seeing or what we were able to identify, was a ribozyme that was 72 nucleotides long that still had activity. And in addition, subsequent 3' truncations of this 72 nucleotide revealed that the actual minimal core was about 44 nucleotides long. And as sort of an aside, there was an interesting correlation between secondary structure and activity. So these are the predicted secondary structures of each one of these constructs. And you can see that the sequences that had the most activity appeared to fold into a similar conformation. And in addition, when you went from 44 nucleotides to 43 nucleotides, the 44 nucleotide or 43 nucleotide ribozyme appeared to fold into a different secondary structure than what we believe is the active conformation. This ribozyme is also interesting because compared to ribozymes found in the original selection, it seemed to have a different TMP and magnesium requirement than that original ribozyme found in the 2014 selection. So in this case, this ribozyme appeared to like excess TMP relative to magnesium, whereas the previous ribozyme seemed to need about 400 millimolar free magnesium. And that was where it was at its optimal. And this kind of suggests that it's possible that this short ribozyme that we identified may have a different mechanism of catalysis than the TPR1 that was identified in the 2014 study. What was interesting when you looked at the secondary structure of our new short ribozyme compared to sequences from Janita's original pool, we were able to identify 3 sequences from that starting pool that appeared to share similar structural motifs. So for example, they all seem to have this 2 nucleotide linker, an identical 2 nucleotide linker that's flanked by these P1 and P2 helices. They seem to have a similar 8 nucleotide sequence on the left hand of the loop, and they all seem to have a similar 3 nucleotide, identical 3 nucleotide bases on the right side of the loop. They differed in the overall sizes of this loop. For example, this sequence has a 4 nucleotide insert here, and this one has a 41 nucleotide insert. In addition, they seem to differ in the extraneous sequences after the 3' end. But we were really surprised to be able to find similar motifs, catalytic motifs, from this starting pool of round 4 of Janita's selection using our method. So as I kind of alluded to earlier, the replication of an RNA world organism, it would have relied on the generation of active nucleoside 5' phosphates. The TPRs that I've been showing here are sort of a proof of principle that RNA is capable of doing this triphosphorylating activity, but its actual utility in an early life context is somewhat limited because it can really only triphosphorylate itself. And so what you really would want is a ribozyme that can phosphorylate free nucleosides. Now that's technically more difficult to do because the product can diffuse away from the ribozyme that made it, so it's harder to tie activity to sequence. So in order to select for ribozymes that can do this trans activity, triphosphorylate free nucleosides, it was necessary to do this part of the selection in emulsion. And so this is work that a previous grad student, Arvind, did in our lab, where he selected in emulsion. And basically the way this worked was you have a randomized N150 pool, and you incubate these pool molecules with 6-thioguanosine and trimethophosphate. And the active sequences were able to bind these two and generate 6-thio-GTP. In addition, the polymerase ribozyme, which is shown on the right here, would then take the 6-thio-GTP and tack it onto the pool molecule that made it. And that way, any active sequences would be thio-modified, and you can use that thio-modification as a handle to fish out active sequences. And crucially, all of this is done in droplets. So that way, you could tie the product to the sequence that created that product. So from the selection, I was able to identify some sequences that could triphosphorylate free guanosines. And the way this assay worked, it's very similar to the selection step, except instead of the polymerase ribozyme tacking on the GTP to the pool molecule, it's now tacking it onto a radiolabeled oligo. And so when you run this on the gel, active sequences you would be able to identify because there's a shift of one nucleotide on the gel. So using this assay, I was able to identify a family of sequences that could use guanosine. And this is important because guanosine is, first of all, more prebiotically plausible. And also, among the 4 canonical nucleosides, guanosine is slower to be triphosphorylated than the other ones. So the triphosphorylation of guanosine would have been very important for an RNA world organism. So from this assay, we took the most active sequences, most active sequence, which is sequence number 59 here. We renamed it GTR1 for guanosine triphosphorylating ribozyme 1, and we took it for further characterization. We used LC-MS to confirm that the activity we observed in the previous assay was indeed due to ribozyme-mediated synthesis of GTP. So what I'm showing here in the top left is the LC trace, and you can see that there's a characteristic peak around 1.8 minute elution time in both the 9 micromolar GTR1 condition and the 6 micromolar control. And there's no peak when you just have buffer. And this fraction corresponded to a mass spec signal shown on the right here, where you have a peak at about 523 m/z, which corresponds to the molecular weight of GTP. And you only see this peak when you have either GTR1 or GTP, and you don't see this peak when you just have buffer. And all of this suggests that the activity we were observing earlier was indeed due to the synthesis of GTP. We wanted to characterize this ribozyme further, so we looked at what the rate enhancement of the catalysis was. So on the left, top left, I'm showing time course in the presence of GTR1, and you can see that it saturates after about 80 minutes, and activity reaches about 100% fraction extended. So the rate is about 1.9 per hour. And then on the right is the negative control with no GTR1. And you can see that it barely reaches above 8% after 700 hours. So that's a rate enhancement of about 18,000. One issue with this ribozyme is the very low turnover number. So we tested different concentrations of GTR1 and calculated the turnover number, and it's between like 1.6 and 1.8. So it appears that the rate limiting step for this ribozyme is the release of GTP after it has been synthesized. It seems like the ribozyme likes to really hold on to the GTP after it makes it. So that's a room for future studies there. We next wanted to, you can imagine, for example, a situation where the replication of an RNA organism is mediated entirely by ribozymes. So for example, a ribozyme like GTR1 could produce the triphosphorylated nucleosides, and then a ribozyme like the polymerase ribozyme could use those activated nucleosides in order to polymerize them into RNA strands. So what we wanted to do was challenge the polymerase ribozyme to use GTP that has been produced by GTR1. So the way this assay worked is we used a polymerase ribozyme with a trans template and primer duplex, and then we fed it ATP, CTP, and UTP, but not GTP. Instead, we gave it the products of a GTR1 reaction. And basically, the idea is any GTP being incorporated should be GTP that has been produced by GTR1. And indeed, we did observe an enhancement of G incorporation in the presence of GTR1. So on the left is our negative node GTR1 control, and you can see that there's very little incorporation after position 2, which corresponds to incorporation of G. And then when you have GTR1 or our positive control of GTP, you see increased incorporation at position 2. I'm suggesting that the polymerase ribozyme is able to use GTP that's been generated by GTR1. So hopefully what I've shown here is that our work in investigating triphosphorylation ribozymes has strengthened the case for the central role of RNA. We know that the RNA world likely was more abundant in shorter RNA. And we also know that the RNA world organisms likely would have required activated 5' triphosphorylated nucleosides. So we were able to show in these two studies that short RNA is still capable of interesting activity in an origin of life context. And we were able to identify a ribozyme that is capable of triphosphorylating free nucleosides. And with that, I'd like to thank our lab and our funding sources, NIH and NASA. And I'll hand it back over.
Thank you, Josh. That was wonderful. So what we're going to do now is actually flip back over to Uli, who will have a bit of a topical introduction for the work that Hannah is doing.
Thanks, Becca. So I believe that the discussion for Josh's talk is also after Hannah's talk. All right. So let me, Hannah's PI, Akif Tezcan, was not able to come today. He's in Europe, I think. So I have the pleasure to introduce him. So Akif Tezcan, oh, sorry, and I should share a slide that introduces what his lab is doing. And here you see a picture of Akif. And so Akif did his undergraduate research at McAllister University, that's in Minnesota. And then he did his PhD and postdoc at Caltech. The PhD was with Harry Gray, and his postdoc was with Doug Reeve. During the postdoc studies, Akif published 3 X-ray structures of nitrogenase that showed the protein conformational changes based on the bound nucleotides, either ATP, ADP, or the nucleotide-free state. And as you will hear from Hannah, this is important for the future research. And in his work at UCSD, Akif also established a second line of research, and that is based on metal-directed protein assembly. And I will show you on the next two slides, on one hand, something about the metal-directed protein assembly line of projects that's going on in Akif's lab, as well as a short introduction to nitrogenase. So Akif was trying to, or he looked for building blocks that he could assemble, and his first building blocks was a modified cytochrome C. And then by adding 3 histidines, and there was already an initial histidine, he generated a total of 4 metal coordination sites. So this monomer with these 4 histidines that can form trimers, dimers, or tetramers, depending on the metal cation that you provide. So if you provide nickel 2+, you form trimers. If you add copper 2+, you form a dimer. And if you add zinc 2+, you form a tetramer. And these ones are all engineered metal-directed interfaces, but you also have metal-directed interfaces for a few proteins in nature. And one of those examples is nitrogenase, and that's the project Akif took over from his postdoctoral research into his lab at UCSD. And on this slide, you see a 3D structure of nitrogenase. So that's a natural example of a metal ion-mediated large protein assembly. And you see these metal clusters here. One is here, one is here, and one is here. And these metal clusters are responsible for catalyzing the nitrogen reduction reaction. And the energy necessary for that step is provided by hydrolysis of ATP. So here you see the reaction. Nitrogen uses the energy of 16 ATPs in order to generate 2 molecules ammonia plus ADP molecules. And of course, one difficulty, or this is a very difficult reaction to achieve, and the conversion of the energy from ATP to the nitrogen reduction proceeds via large conformational changes in this protein. And of the metalloclusters that you see here, 2 of the 3 are unique to nitrogenase due to their stoichiometry and their size. And with that, I think I've said enough about nitrogenase. And please welcome Hannah Rutledge, who's 6-year student in Akif Tezcan's lab.
And then I'll also introduce you with your bio quickly so we can hear that. So we already heard that you're in the testing group, but so also prior to attending grad school, Hannah was a high school chemistry teacher. She received her BS from Rice University, where she conducted research on quantum dot solar cells, excuse me. Currently, she is studying electron transfer and nitrogenase with a focus on protein dynamics. And as we heard, she is investigating conformational changes both on a large scale, the whole enzyme complex, and on a smaller scale, the electron transfer relay, the PCluster. Take it away.
Thank you for the introduction. As mentioned, I'm Hannah. I'm from Akif Tezcan's group at UCSD. And today I'm going to be talking about nitrogenase and specifically one of the clusters in nitrogenase called the PCluster and how it evolved to be redox metastable. And to begin with, I like to take a look at the whole nitrogen cycle. And so this non-industrial nitrogen cycle, it's the biogeochemical cycle of nitrogen transformations on Earth. And so if we think about this entire cycle, perhaps the hardest step in it, chemically speaking, is actually the nitrogen fixation step. And this is where nitrogen becomes bioavailable by reducing atmospheric nitrogen into ammonia, which is a bioavailable form. And so when we think about nitrogen fixation and these reduced forms of nitrogen, they're necessary for life. And the reason they're necessary for life is because many biomolecules, such as proteins, which have reduced forms of nitrogen in their backbones, along with some of the side chains, are needed, along with many, or all nucleotides for RNA and DNA also require reduced forms of nitrogen. And so if we think about today, in this day and age where we get our reduced forms of nitrogen, as humans, it's from the food we eat. We cannot take atmospheric nitrogen and convert it into these reduced forms of nitrogen. And so in the food we eat, the reduced forms of nitrogen mostly come from two places. One of them is industrial nitrogen fixation, which is done by the Haber-Bosch process. And in industrial nitrogen fixation, we combine molecular nitrogen with molecular hydrogen over a catalyst to get ammonia. And one thing that I like to point out here is this is a very difficult process. The triple bond in nitrogen, it's nonpolar, it's very strong, it's hard to break. And we also want to favor product formation. And in order to achieve this, Haber-Bosch process uses extreme pressures and extreme temperatures in order to favor ammonia production at an appreciable rate. On the other hand, there's also biological nitrogen fixation. So this is carried out by diazotrophs that have the enzyme nitrogenase. And in this reaction, we combine molecular nitrogen, not with molecular hydrogen like in Haber-Bosch, but with protons and electrons. And as mentioned by Uli, this is all powered by ATP hydrolysis. And what's interesting about this is it can occur at atmospheric pressure and room temperature. And so there's a lot of interest in understanding how nitrogenase works, perhaps someday to develop a more efficient and sustainable industrial process to mimic it. But if we think about nitrogen fixation and biological nitrogen fixation specifically as we know it today, it hasn't always been like this. And so in terms of the evolutionary history of nitrogenase, at some point in early Earth, fixed nitrogen was most likely limiting. And so, organisms have these selective pressures now in order to develop biological mechanisms for nitrogen fixation. And if we want to think about how this evolved, we should start by thinking about where was this most likely to evolve. And as mentioned for Haber-Bosch, we're using extreme temperatures in order to help break this triple bond. We also want an environment where products such as ammonia can be quickly displaced in order to drive product formation. And so the most likely place that scientists have hypothesized this occurred was at thermal vents, where there's a lot of heat. There's also minerals such as iron that would probably be required. And there's actually a small amount of abiotic nitrogen fixation that can occur near these thermal vents already, perhaps providing some amount of nitrogen already in fixed forms. And in terms of a timeline, if we think about this, the organisms near these vents oftentimes are methanogens. And methanogens, they already have an enzyme called CFBD, and this is a nitrogenase homologue. And so we think that the proto-nitrogenase actually developed from CFBD in these anaerobic thermophilic methanogens near these vents. And so this isn't the nitrogenase we know of today. Part of the reason we know that is because if it evolved from CFBD, this proto-nitrogenase likely was some sort of homooligomer. And as I'll show you in a slide coming up, the nitrogenase that we have currently in today's world is not a homooligomer, it's a heterooligomer. Around 2.5 billion years ago, we had the evolution of our oxygen atmosphere because of oxygenic photosynthesis. And sometime after this, we think there was a gene duplication event where the molybdenum nitrogenase that I study actually came about, and the NIF operon that has all of the nitrogenase components, it has to have NIF D, K, E, and N, plus some other things. But we think this is around the time that this operon came about. And then finally, about 1 billion years ago, we think that there was horizontal gene transfer and nitrogen fixation then transferred into aerobes. And as you'll see, there's a lot of metal clusters. They're all very oxygen sensitive. So this also required lots of oxygen tolerance mechanisms to evolve as well. So the question is, in this day and age, what does nitrogenase currently look like? And so it's a pretty massive enzyme, and the entire complex is 360 kilodaltons. And what I'm showing here is half of the complex because it's symmetric, and so this is how I'll depict it in the rest of my slides. And it's composed of 2 component proteins. In green at the top, we have what's called iron protein. So this is where ATP hydrolysis is occurring, and it's also the biological reductase of the catalytic portion, which is called the MOFI protein, shown in blue and purple. And so this blue and purple protein, it's a heterotetramer, And this is what we think evolved from the CFBD in methanogens, which initially we think was some sort of homodimer or homotetramer. And so how does nitrogenase actually function? What do we know about it right now? So we know that the nucleotides bind to iron protein, and then there's 3 metalloclusters. And so nitrogenase is doing the biological reduction of dinitrogen, and it requires many electrons to transfer. And so these clusters act as a molecular wire through which electron transfer can occur. And the first cluster, which is in the green iron protein, is a 4-iron 4-sulfur cluster. And then we have 2 more clusters that are in the catalytic portion of MOFI protein. We have the P cluster, and then we have the active site FeMo cofactor, or FeMoCo for short. And what's really interesting, what Uli alluded to, is that 2 of these 3 clusters are unique to nitrogenase and not found anywhere else in biology. And that's the P cluster and FeMoCo. Plenty of other metalloenzymes in biology have 4-iron 4-sulfur clusters and smaller iron-sulfur clusters as well. But the P cluster and FeMoCo, they're quite large. They're energetically very expensive for the cells to make and to synthesize and insert into MOFI protein. So clearly they have a unique evolutionary role in nitrogen fixation, and they're required by these cells. If you look at other nitrogenase homologs, they tend to have a 4-iron 4-sulfur cluster in place of the P cluster. So MOFI protein is very unique in having this P cluster. It's not surprising that FeMoCo is a unique cluster because it's responsible for the binding and activation of dinitrogen, which is very difficult. But for me, when I started grad school, I thought it was very interesting that the P cluster would be unique, and it's not just a 4-iron 4-sulfur cluster. And so I was very interested in understanding why this intermediary electron relay site would be an 8-iron 7-sulfur cluster and what sort of roles it might play. And so in order to understand this, we have to think about how does electron transfer happen in nitrogenase. And so we think that electron transfer is conformationally gated. And what this means is that in order for electrons to pass through these clusters, this molecular wire, conformational changes must occur within the enzyme complex. And there's a lot of circumstantial evidence for this. So first of all, iron protein, the green reductase, it's the only reductant that can activate MOFI protein for any significant amount of dinitrogen reduction. If you throw a stronger small molecule reductant at MOFI protein, you won't get any nitrogen fixation. You have to have iron protein. So clearly, the protein itself, not just its reductive power, it is having specific interactions with the catalytic MOFI protein, enabling electron transfer to occur. Second of all, we also know that we have what's called the deficit spending model of electron transfer. And what this is if you look at this, you might intuitively think you start with electrons at your 4-iron 4-sulfur cluster, they jump to the P cluster, and then they end up at the active site FeMoCo. But what actually is happening is the first electron transfer starts from the P cluster and goes to FeMoCo, creating a deficit of electrons in the P cluster, which is then backfilled by the 4-iron 4-sulfur cluster's electrons. Yet, even though this first electron transfer happens within MOFI protein, it can't occur until iron protein binds. So we think that iron protein is transducing conformational changes within MOFI protein that allow that electron transfer to occur by opening our conformational electron transfer gate. And then thirdly, there's different docking geometries of iron protein on top of MOFI protein dependent on the nucleotides bound. So for Akif's postdoc, he crystallized the nitrogenase complex in 3 states. On the left is the nucleotide-free state. In the middle is a non-hydrolyzable ATP analog. And on the right is ADP. And what you can see is over the course of ATP hydrolysis, iron protein rocks from the left side of MOFI protein to the center to the right. And at some point during this rocking and these conformations, you can think that there might be conformational changes induced in MOFI protein that allow this electron transfer to occur. And so the question is now, what are these conformational changes? Where is the conformational gate? And the answer is we still have no idea. If you overlay all of the structures of MOFI protein on top of each other, they're perfectly superimposable with the exception of some conformational changes in the clusters. And so the P cluster actually has some large, like metallocluster standards, conformational changes. In Azotobacter vinelandii, or AV, the model organism for studying nitrogenase, the P cluster in the fully reduced state as isolated looks like 2 4-iron 4-sulfur clusters fused at a central sulfide. and it's bound by 6 cysteine residues. Upon 1 electron oxidation, you actually have a serine that kind of swoops in here and binds one of the irons, pulling it away from the central sulfide. And so this 1 electron oxidation has quite a conformational change of this cluster. Serine ligation to clusters is also pretty rare, so this is pretty unusual. Upon a 2nd electron oxidation, the backbone amide of one of the bridging cysteines comes in and binds another one of the irons. And so this Azotobacter P-cluster has these large, it's also reversible, redox-mediated conformational changes. And this was discovered in the 90s with these crystal structures. However, it was thought it probably isn't that important. It's probably not relevant to catalysis. And the reason people thought this is because while a lot of organisms do have that serine in position 188, a lot of other organisms only have an alanine in that position of their nitrogenase. And alanine cannot bind irons of iron-sulfur clusters, so clearly, right, this must not be important. But on the other hand, there was a postdoc in Akif's lab, Cedric, who decided to crystallize nitrogenase MOFI protein from one of the organisms that has alanine. And so this organism, Gluconacidobacter diazotrophicus, he purified it, he crystallized it, and what he found was the P-cluster in the reduced state, it looked pretty much just like Azotobacter, only instead of having a serine here, now we have an alanine. However, upon 2 electron oxidation, there's another oxygen-based residue. Instead of a serine, it's a tyrosine this time, and it's in a different position, but it comes in and it has an analogous binding to the P-cluster where the 2 irons open up away from the central sulfide. So just like Azotobacter, Gluconacidobacter's P-cluster also undergoes a large reversible redox-mediated conformational change. And so the next step was Cedric looked at a bunch of different sequences, and what he found was there was a covariance. So organisms that have the serine in position 188 tend to lack the tyrosine, and then organisms that have the tyrosine in position 99, like Gluconacidobacter, tend to lack the serine. And out of 95 nitrogenase sequences, this is true for 92 of them. In 2 of the sequences, they actually have both serine and tyrosine, and then there's only one that has neither. So this tends to be pretty conserved to have this oxygen-based ligand around the P-cluster that can come in and bind. But the question is, well, we know it's there, but why do we need it? And so when I started grad school, I decided to investigate this ligand. And so I made 3 P-cluster mutants in Azotobacter, and I characterized them. And the 3 mutations I decided to make was I made a serine to alanine mutation, in which case the P-cluster has no oxygen-based ligand, neither serine nor tyrosine. I made a double mutation, so it has that alanine, but I also inserted a tyrosine like Gluconacidobacter. I call this my Gluconacidobacter-like mutant, so it has tyrosine instead of serine. And then lastly, I made a mutation in which my Azotobacter mutant had both serine and tyrosine, so this is F99Y. And so I'm going to tell you a little bit about these 3 mutants, and then some follow-up studies that I've been doing on one of them that's been very interesting. And so perhaps most interesting is the S188A MOFI protein. And so S188A, this is an Azotobacter. It has an alanine instead of the serine, and it has phenylalanine naturally, so no tyrosine. So there's no oxygen-based ligand around the cluster. And in this crystal structure, and in all the ones following, I'm actually showing anomalous density maps, these black mesh maps. And this is showing exactly where the irons are located within the cluster. And in this fully reduced state, the reduced S188A mutant looks pretty much just like wild-type Azotobacter, other than the fact that there's no serine, there's an alanine. What's really interesting is upon 2 electron oxidation, you have complete loss of 2 of the iron centers. So previously, we'd seen that in wild-type, the serine bound one of the irons, and then the backbone amide bound another iron, and the cluster opened up. However, here we have complete loss of 1 iron from each of the cubanes, for a total loss of 2 irons for the P-cluster. And so, of course, I think naturally my next question was, well, can we put irons back in? And being a good grad student, I thought, what's the easiest possible way to try this? And I thought, I'll just re-reduce the cluster. I'm not going to give it any more iron the first time I try this, and I'll just see if it can pick up iron from anywhere. And sure enough, upon re-reduction, I had full reconstitution of the cluster. And I think what happened was I was actually cannibalizing other protein in solution for its iron content, and then what crystallized out was the fully reconstituted protein. And so what we see now is serine actually can protect the P cluster from redox lability in Azotobacter. The next mutant that I looked at was the double mutant. This is F99Y S188A, and this is my Gluconacidobacter-like mutant. So it has tyrosine in place of the phenylalanine, but no serine. And just like before, in the fully reduced state, it looks the same as wild-type Azotobacter, which looks the same as reduced S188A, which looks the same as reduced Gluconacidobacter. And upon 2-electron oxidation, we see a binding of the tyrosine to the iron and the backbone amide. And so this at first glance looks just like the wild-type Gluconacidobacter. But upon further refinement of the crystal structure, what I found was that these 3 iron centers circled in red are all partially occupied. And so in total, on average, I lose 1 iron per P cluster. So even though this looks like Gluconacidobacter, it's not as stable as Gluconacidobacter's P cluster. So I think Gluconacidobacter has other amino acids that it evolved in order to help stabilize a cluster that are not in this primary coordination sphere. And I think that the tyrosine, in this case, is actually kind of serving to shuffle the irons around in this equilibrium to allow this 1 iron loss from each P cluster. And then just like with S188A, upon re-reduction, I actually fully reconstitute the cluster and all of the iron sites are fully occupied again. The last mutant that I looked at was F99Y. So this mutant has both the serine and the tyrosine. And I wasn't sure if I'd see both of these ligands binding or only one binding upon oxidation. And what I found was that when it was oxidized, the Azotobacter P cluster just showed serine ligation in the crystal structure. And this isn't terribly surprising because Azotobacter evolved with the serine. Gluconacidobacter clearly evolved with the tyrosine and has other amino acids helping stabilize it. And then after I got these crystal structures, I wanted to further characterize these P clusters. And so luckily, because they're metal clusters, I can use inorganic techniques such as EPR to look at them. So EPR, you can look at paramagnetic unpaired electrons. And if you have an electron, it can have an upspin or a downspin. If you put that electron into a magnetic field or a microwave field, you can actually split those levels and they become degenerate. And so then if you take it and you sweep a magnetic field over it, you can hit the resonance and you can see an absorption at that resonance corresponding to the split in the energy levels of that electron. And of course, this is a very simple case here that I'm depicting. However, with nitrogenase, we have multiple unpaired electrons and it's anisotropic. So our EPR spectra get a little bit more convoluted. But what I'm specifically looking at are signals that are going to be arising from the P cluster. I also still have FeMoco in there, so we're going to see some FeMoco signals as well. So here, what I'm showing is all of these are the reduced mutants and wild type. And in the reduced state, the P cluster is actually not paramagnetic. So the P cluster in the reduced state is EPR silent. So you still see all these EPR signals in the wild type F99Y, my Gluconacidobacter-like mutant, and my S188A mutant. And all of these signals are from FeMoco, which is why they all look the same, because we haven't done anything to change FeMoco. However, upon oxidation, what's really interesting is that on the top, in the green and the teal, are my 2 mutants that have a labile P cluster, and they both have new signals arising in the G2 region. And so to begin with, in teal, this is my Gluconacidobacter mutant, Gluconacidobacter-like mutant that lost 1 iron per P cluster. And what we see here is it has a really strong signal. And this signal is actually very similar to the 1 electron oxidized P cluster that has been previously published. And so how I interpret this is that each of these P clusters, it's not losing an iron 2+, it's losing an iron 3+. So I'm losing 1 ferric iron, and I have what I call a 1 electron oxidized P cluster, even though I don't know if you can call a 7 iron cluster the P cluster, but I think that's what's going on here. And then in S188A, I have that same signal, it's just much weaker. So I think I still have a small population of P clusters that haven't lost 2 irons, but only lost 1. On the other hand, in wild type and F99Y, we don't see those signals. And the P cluster, while it is paramagnetic in the 2 electron oxidized state, doesn't have a signal typically in perpendicular mode EPR. And that's because you're actually looking at forbidden transitions. And so in order to see these forbidden spin transitions, you actually have to look in parallel mode EPR. And so what's been known for many decades is that for wild-type Azotobacter, when you oxidize it, it has a unique signal at about G12 in parallel mode. And people aren't quite sure what's going on with this. It's often been said as just a phenomenological signal. And what's interesting is if you look at wild-type Gluconacidobacter, its signal shifts to G16. And so there's a hypothesis for a long time that this is just helping us to look and see what's the identity of the oxygen-based ligand. And perhaps if you have serine, you have the G12 peak, and if you have tyrosine, you have the G16 peak. And what's interesting is my mutants support this hypothesis. So in my mutant that has both serine and tyrosine, we mostly have the G12 signal, as we saw in the crystal structure with the serine ligation. But we also have a small signal at G16. And I know it looks like noise. This was over 100 scans. It's definitely a real signal. The signals are weak because these are forbidden spin transitions. But this tells me that there is a small population that might have the tyrosine bound. And in my Gluconacidobacter-like mutant, I've totally gotten rid of my G12 signal, but now I have the G16 signal, like wild-type Gluconacidobacter. And when I have neither serine nor tyrosine, I'm EPR silent in parallel mode. So this helps us now identify the oxygen-based ligand that's binding to the PCluster. But there's always a but. When I first studied the activity of these mutants, I saw that in terms of nitrogen reduction in vitro, all of my mutants were the same activity as wild type. So it doesn't really matter what color is what here. They're all pretty much overlaid with each other, and there's no significant difference between wild type's nitrogen reduction ability and any of the mutants, regardless of whether or not they have a labile PCluster. So I was kind of wondering what's going on here. Does the serine actually matter? And then furthermore, if you look at the growth curves of the mutants along with wild type, and these, just to clarify, all of these mutants are made in Azotobacter. They're not overexpressed in E. coli. And when I make these mutants, the way I express them is I starve my Azotobacter cells of fixed nitrogen, so it forces them to express nitrogenase in order to survive. And so under nitrogen fixing conditions, all of these mutants grew just as quickly as wild type. And it made me wonder, do these labile PCluster mutants grow as quickly just because I'm giving them, optimal conditions to grow in? So when I grow these mutants, I'm actually growing with a ton of iron in the media. So I'm growing with 35 micromolar iron. And so I wondered, maybe if they're losing iron during catalysis, and if it's somehow harming nitrogen fixation in vivo, even if I didn't see it in vitro, maybe if I knock down the iron concentration, I'll slow down their growth rates. So what I did was I took S188A, the mutant that loses 2 irons per PCluster upon oxidation, and wild type Azotobacter, and I grew them with much less iron. So with my normal growth conditions, what I call 100% iron, 35 micromolar, you can see they have the same growth rate. However, when I knock that down to 1% of the normal iron that I used, so only about 0.4 micromolar iron, wild type's growth rate does decline, but what's interesting is S188A's growth rate is even slower than wild type. So now I can distinguish them based on growth rate. And this tells me that somehow S188A is being hindered by that mutation. And I've raised a few more questions now. So first of all, under 100% iron conditions, are they growing at the same rate because they have the same activity? Or are they growing at the same rate because S188A is overexpressing its nitrogenase and upregulating it compared to wild type? And then maybe when I knock the iron concentration down to 1%, maybe S188A can no longer upregulate its multi-protein production because there's not enough iron in the cell, or maybe the activity is less because there's not enough labile iron around to reconstitute the cluster during catalysis. And so I decided to do a few studies to look at the expression rates of these proteins. And so what I did was I did RT-qPCR to measure the amount of NIFK transcripts. So NIFK is one of the subunits of MOFI protein. These are technical triplicates of biological triplicates. And what I found was under 100% iron, so my normal growth rates with optimal conditions, both wild type in blue and S188A in green, had the same MOFI protein expression levels. However, under 1% iron, when I really knock it down, wild type, its expression drops slightly, which might just be because of iron limitation. It's working on other things in the cell. However, S188A's expression suddenly jumped up, and now all of a sudden we have an appreciable difference in the amount of MOFI protein being expressed. So my next question was, well, is this because somehow the activity is changing when there's less labile iron around? So I conducted in vivo nitrogenase activity assays, where actually instead of looking at nitrogen reduction, I looked at acetylene reduction by whole cells. And then using the amount of MOFI protein transcripts, I could then calculate out the specific acetylene reduction activity, or sorry, relative acetylene reduction activity. And what I found was under 100% iron conditions, wild type and S188A had the same nitrogenase activity. However, under 1% iron conditions, all of a sudden S188A's activity is much lower than wild type. So this tells me that perhaps I actually am losing iron during catalysis, and that the serine ligand actually does play a functional role during nitrogen fixation. And so in summary, the whole question I set out to answer, what are the roles of serine 188, serine 188 in Azotobacter MOFI protein? And I found 3 roles. The first is that it provides redox metastability to the P cluster, as we saw in the crystal structures, and allows the P cluster to be oxidized without losing its iron. It also is required for efficient growth in iron-limited media. And this is important because in the environment, you're not necessarily going to have all these diazotropes have tons of iron around. And lastly, I didn't talk about it in these slides, but I've also seen that it prevents mismetallation of the P cluster during oxidative stress in vitro. And so with this, I'd like to thank the group. So I'd like to thank everybody, but especially Akif. And then I've had some wonderful undergrads that have helped me on some of these projects. So Alkane, Laura, and Robin, along with some collaborators, had Mackenzie and Dr. Mike Green at UC Irvine helping out with EPR. And then Dr. Dennis Mee at Virginia Tech has kindly provided some Azotobacter strains for us. And so I'd like to say thank you.
Thank you so much, Hannah, and all of our speakers. I'll give a round of applause on behalf of our audience who can't do that. But yes, thank you so much. We are going to now roll into the discussion portion of today. So yes, I see you're all back online. We're going to open up the chat. So as the audience, you can raise your hand, and I will do my best to acknowledge you and get your mic turned on, or if you prefer, I can read your questions through the chat, either way. And also remember, if we don't get to your questions today, we do have Slack for continuing to talk after the event. So with that, we can go ahead and jump in. I think we're still opening the chat. Quick question to co-host James. Is the chat open right now? Sorry, technical difficulties.
I think the chat is open now. I think I saw at least one pop up now.
Awesome. So there is a question for Josh from Vikas Nanda, excuse me. Do you have some idea about the evolvability of your ribozyme? How big is the sequence space for availability?
It's a good question. For the TPR ribozymes, we have some idea because from Janina's original selection, they were able to identify hundreds of these ribozymes by the end of the selection. So we think that those types of ribozymes are fairly common. The guanosine triphosphorylating ribozymes may be more rare just because we're challenging the RNA to do more things. It has to bind 2 different things and then put them together in trans. So the available sequence space of a ribozyme may depend on the type of catalysis that you're challenging it to do. And what was the other question? Yeah, how big is the sequence space for activity? Yeah, I would just say it depends on the type of catalysis.
Josh, I think the question goes a little bit to what the ribozyme you have right now can evolve to in the future. How can you improve it? And I think this is how the question was meant.
Okay, yeah. We definitely want to improve the turnover. And so in terms of evolvability, we're definitely planning to do some either reselections or just engineering with that particular ribozyme. We have some data to suggest that some of the elements of that particular ribozyme may not be necessary for catalysis. So there's a helix in that ribozyme that we think we could truncate, for example, which would mean that the sequence requirement may not be as stringent. We might be able to remove, for example, 20 nucleotides, which would lower the sequence ribozyme or sequence requirement for that type of ribozyme.
Yeah, I think the bottom line to that question is we just don't know. We are just in the very beginning of that.
Yeah, but we're working on it.
So Uli, you can go ahead and ask your question.
So yeah, I was interested in the evolutionary origin of your nitrogenase, and you showed that the methanogens may have come up with the earliest stages. Can you say some more? Or was it the iron 4 sulfur 4 cluster that originated earliest, or was it the other ones that originated earliest?
So initially, I mean, there isn't too much known about this. That methanogen protein that we think was a starting point for nitrogenous evolution. It wasn't until just a couple of years ago that people even knew what its function was, and it was just called a NIF-like protein until a few years ago. But for the proto-nitrogenase, we don't know for sure what clusters it contained. We do think that it was a homo-oligomer, which probably means it didn't have the P cluster. It probably had maybe a 4 iron 4 sulfur cluster in place of the P cluster. And then Fimoco definitely wasn't what we know of it as today, and it didn't have molybdenum, which I'm going to kind of answer a few questions at once because somebody also asked about how the iron sulfur clusters are synthesized. And so in the NIF operon, there's actually a whole host of other enzymes that are required for the synthesis of both the P cluster and Fimoco. So Fimoco requires a radical SAM enzyme that didn't evolve until much later. So we know that the proto-nitrogenase definitely didn't have Fimoco. It had probably a pre-Fimoco-like complex that just had iron and sulfur and no molybdenum. But there are many other enzymes, and this is part of the reason that we don't express nitrogenase in E. coli, is because E. coli can't handle all of the scaffolds and other machinery required for the synthesis of these clusters. I'm not sure if that was specific enough to answer both your and Thomas's questions. But.
Yeah, thank you.
And I think that also may have answered Dragos's question as well about which of the metal clusters were evolved and incorporated first. So
yeah, I don't think it's really known quite yet which one evolved first, because you had to have the gene duplication for the 2 different subunits, and then also the NIF E, N, and B for the Fimoco synthesis. And I don't think the order of that evolution is really known at this point.
Something to find out still, right? So we have a question from Kevin Devine for Josh. How efficient are your ribozyme kinases at phosphorylating adenosine, uridine, and cytosine?
Yeah, that's a good question. I haven't tested that myself. I'm not sure if Arvin ever tested the triphosphorylating ribozyme with the other 3 nucleobases. So again, we don't know, but we're definitely going to try to evolve ribozymes that would be able to triphosphorylate each of the 4 canonical nucleobases.
And one thing that can partially address this question is that, so Josh did the screen for the different clones with guanosine and also with 6-thioguanosine. Remember, 6-thioguanosine was used during the selection. And what I found striking is that there is a huge difference in activity, which cluster is best with guanosine and which cluster is best with 6-thioguanosine. So we do expect that there are dramatic differences with the adenosine, cytosine, and uridine.
I believe we have a follow-up. Sorry, I'm trying to unmute and hitting buttons. So I think we have a follow-up from Kevin on that same question. Kevin, are you able to ask that question?
Yeah, can you hear me okay? Yeah, thank you once again for 2 excellent presentations. So question for Uli and Josh. Have you tried looking at ribozymes with, some of the non-standard nucleobases that you see in transfer RNA? For example, 3-carboxypropyluridine and 5-methylaminomethyl, thiouridine, because one of the problems of ribozymes as opposed to proteins is that they're much less efficient at doing catalysis. That's down to the fact that the amino acid side chains give us a much richer source of reactivity through the more diverse functional groups. So has anybody tried to look at that with ribozymes with bases which contain additional functionality?
Josh, you want to answer? The answer is simple. It's a no.
Yeah, we haven't tested it.
Yeah, basically this ribozyme is in its very early stages. So anything that Josh showed you about this ribozyme is just under review right now. So we just have finished this study far enough that it can be published, but we haven't done much further experiments yet. But what you describe, I think, is an excellent idea, that we could explore the substrate spectrum that this ribozyme can take with many different modified nucleosides. And our long-term goal is something, as Josh alluded in the experiments with the polymerase ribozyme, the long-term goal is to make a system where when NTP synthetase can provide sufficient NTPs so that a polymerase ribozyme can make polymers with it and efficiently maybe self-replicate. For that, I think our main focus is currently on making these ribozymes better for the 4 canonical nucleotides. But in order to understand how they work, I think it's an excellent idea to also use those modified nucleotides and NTPs.
Yeah, I mean, I was obviously thinking more of, the things with additional bases with more richer functionality, they'd be more useful as ribozyme nucleases, perhaps, rather than kinases, I was thinking. Because with additional functionality, they'd like
I think your line of thinking is probably correct. There were probably non-canonical bases that existed, at the beginning of the RNA world. And these bases may have enriched the functionality of ribozymes early on. I don't think we're at the stage yet to start injecting some of these non-canonical bases into our ribozyme yet. Like Uli said, we're still early on in development. We don't even know where the catalytic site is or the binding sites are. So, but that's definitely something we can look into.
Thank you.
Kevin, would you like to go ahead and ask your question as well for Hannah?
Oh, yes. So, Hannah, have you tried looking at replacing the essential serine and tyrosine residues with threonine? You're replacing a phenolic OH and a primary OH with a secondary one.
That's a good question. So to give a little bit more background on these mutations and expressing azotobacter, making and expressing and purifying a mutant takes months for nitrogenase as opposed to like a week when you're using E. coli expression systems. So I've been very conservative with my mutations that I've done. And then the way I actually screen for my mutants, which luckily all of my mutants have been able to fix nitrogen, is I test to see if my cells can fix nitrogen. So I have made the S188T mutant, and I know that it can fix nitrogen, but I haven't actually purified it or characterized it at the moment. For the Y98T, are you referring to an azotobacter or are you in gluconacidobacter? So the organism that natively has the tyrosine?
Mm-hmm. Yeah, as well. Which the first one, the first one you mentioned.
So, so that's something else that's been a little bit difficult. Is I have to alter the genome. I'm not actually doing plasmid expression, and I have not figured out a way to alter the genome of gluconacidobacter in order to do mutations and get gluconacidobacter multiprotein mutants. I've kind of been working on that, but it hasn't gone too far. So I don't have a Y98T mutant, although I have been very interested in making those types of mutations in gluconacidobacter. But I think that's something further down the road once we get the microbiology working within gluconacidobacter.
Okay, thank you.
Thank you.
Well, I can ask a question. So I have a question for Josh. So you did do that experiment where you took the product of your GTP forming reaction and put it in with the polymerase ribozyme. I didn't quite catch whether the GTP producing ribozyme was still in that solution when you added it in with the polymerase. And if it was present there, would you expect it to form any sort of complex with the polymerase or affect its fidelity or the reaction in any other way?
Yeah, that's a great question. We do have some evidence that the triphosphorylating ribozyme does, in the selection, it does directly interact with the polymerase ribozyme. So I think your line of questioning is correct. After the GTR reaction, we actually inactivate it with a DNA inactivator, and that completely abolished this activity and also unfolds it. So it likely doesn't, it no longer interacts with the polymerase ribozyme. It is still present in the polymerase ribozyme reaction, it's just inactivated, to answer your question.
Yeah, I think it's really interesting. Uli mentioned this a little earlier, how these things might all work in concert in some sort of grand reaction at some point.
That's what we are trying to do. The long-term goal is to make an RNA-mediated self-replicating system, perhaps with a few cofactors, and replicate an early stage of life.
Are there, okay, we have a question from Eliza. I think you should be able to unmute now.
Yeah, thank you. Yeah, along those lines that Rebecca was mentioning, I was wondering, isn't it your slow release, so slow turnover, isn't it in contrast with the contrast with the fact that your polymerase actually can add the triphosphate and can use the triphosphate. So the ribozyme is working with the polymerase, but it has to give it up a little bit, right, to add it to the growing chain. So that's one question. And the other one would be, if still along those lines, wouldn't it be useful to add maybe a step in the selection where you are allowing the ribozyme to release the GTP when it's not triphosphate anymore, or the contrary, you know, when it is triphosphate, sorry. I'm done.
Yeah, I think for your first point, it is true that in the selection, the any potential GTRs need to give up the GTP. I think part of what I was alluding to with Becca's question was that the polymerase ribozyme could potentially interact directly with the GTR1. So it's probably stealing the GTP from the GTR1 during the selection. It's also true that not, like 100% of the ribozyme isn't holding on to the GTP. It's probable that some of the GTRs are giving up the GTP to the polymerase ribozyme. And what was the second part of your question?
I was just wondering if you had any plans on adding some steps to the selection that would allow you to actually select for higher turnover. And by the way, great work done on it. It was really nice.
Oh, thank you.
Yeah, it's very interesting. And we do have, the expanded genetic alphabet whenever you want to go that route.
Oh, great.
Publicity, right?
Sounds like a great collaboration.
Right. I was just wondering about the, if you were thinking of adding extra steps. I don't know. PNP riboswitch thingy. I don't know, anything.
Oh.
No,
yeah, I think we don't have any plans to modify the in emulsion selection, but I don't know how much we want to get into this, Uli, but we are planning a collaboration with another lab to continue evolving.
So I think Lisa has outlined our next maybe 7 years of research on this ribozyme very easily, because on one hand, with your first question, you describe what we are currently trying to do. We're trying to understand the ribozyme a little better, and perhaps with a few mutations, make it capable of multiple turnover catalysis. And the other thing, that depending on how well the first thing goes, we may want to reselect. And then you're exactly right. We want to put selection pressure on producing more NTPs. And one of the ways how we could do this is if we request or require the ribozyme to make many GTPs or many different NTPs before it would get selected. It's all in the books, and well, we are.
Yeah, that's great.
If we would be able to do this, I think we would be a big step forward. And maybe if the seminar series is still existing in 5 years, we can give you an update.
Maybe 3 years. You guys are good. Yeah, it's gonna happen. All right. Thank you so much for the answer and for the great talk.
Thank you for the question.
And of course, you're welcome back here in the future to give us that update. I have a bit of a follow-up on that. So do you expect that for your ribozymes, you would end up finding one ribozyme that is promiscuous and can do all the different nucleotides, or a family of closely related ribozymes that are each individual and specific?
I think that in the context of early life, you would probably want a more promiscuous ribozyme that can initially decorate each of the canonical bases with a triphosphate. And then you could take that starting point and evolve it to be more efficient. Yeah, I think that's what I would think would be most useful for an RNA world organism.
And then the other part of the question would be, what do you think that our currently existing guanosine triphosphorylation ribozyme could most easily do? Do you think that can be modified into one that is promiscuous and triphosphorylates all kinds of different nucleosides? Or do you think we just need to make 4 different nucleoside triphosphorylation ribozymes so that they can do the job together?
I think we'd have to test it. I think we need to test our ribozyme with adenosine, uracil, and cytosine, and see see what its activity is with those three and go from there. If it's non-zero, I think it would be worth it to try to evolve it towards those other three bases. If it's completely flat and zero, I don't know, it might not be able to ever do that unless you completely, if you do like 30 selection steps, I don't know.
And one additional thought, maybe, when you think about what's most useful for an RNA world organism, I imagine that there were lots of different nucleosides around. So as Kevin Devine said, we might have had those amino acid modified nucleotides or lots of different nucleotides. And at some point, the organism needs to be selective and make only the canonical NTPs. Otherwise, it's going to waste a lot of energy on metabolizing other NTPs, and then it's going to have difficulty on making strands that are either pure RNA or pure DNA or a pure of something else. So I think at some point you need selectivity. So there is a danger with promiscuity too. On one hand, there would be a benefit of having one ribozyme that does it all. On the other hand, if you have one ribozyme that does it all, that same ribozyme may also be more prone to triphosphorylate other nucleotides that you don't want to have.
So we had a question come in for Hannah from Elva Escobar, saying first of all, he enjoyed all presentations, and then he wonders on the role of environment, nitrogen, oxygenated and anoxic environments, what their role is on the molecule evolution.
So that's a really interesting question, and it's still pretty debated. So if you think back to the equation that nitrogenase is catalyzing, whether or not it requires 16 ATPs or perhaps 8 is still debated, but it requires a lot of energy to reduce dinitrogen in the form of ATP. And in addition, it requires pretty hot electrons for this reduction. And so this kind of created this paradox where initially scientists were thinking, nitrogenase must have evolved after, respiration. That way there was the energy provided for everything. But in fact, it evolved prior to then. So there's this paradox of how did they get enough energy, which we think is why they evolved near the thermal vents. But then you also have the oxygen dilemma, where if you do have respiration to provide the energy for nitrogen reduction, you also have the dilemma of oxygen kills nitrogenase, it destroys the clusters. And so there's been a lot of mechanisms that aerobes that are diisotrophs have evolved in order to protect nitrogenase. There's two main forms. There's conformational protection, in which there's actually some proteins that will bind to nitrogenase in the presence of oxygen and temporarily disable it while protecting the clusters. One of these is called the Schiessner protein, and there's a crystal structure of that published a couple of years ago that's pretty interesting. But then there's also respiratory protection, which has evolved. So the organism I study, Azotobacter, is an obligate aerobe, yet it fixes nitrogen in the presence of oxygen. And the way Azotobacter gets around this is it has the highest known rate of respiration. So it's actually controlling how much oxygen is in the cytoplasm by just respiring a lot and limiting that amount of oxygen. And so there's definitely a lot of mechanisms involved. If you look at NIF operons between aerobes and anaerobes, they do have different proteins within them, other than the pretty conserved NIF, EN, B, H, D, and K. And they also have different mechanisms for actually expressing nitrogenase as well.
Thanks, Hannah. We are at time, but there is one quick question hanging in the chat for Josh, so we'll just get to that one if that's okay. It's from Juliet Langleis. I'm sorry, people, with my pronunciations, but the question is, how specific is your ribozyme? Do you obtain only the 5' GTP as a product and only with trimetaphosphate? Thank you both for the presentations.
Yeah, I think according to the mass spec data, it appears that the 5' GTP is the major product. We know that the ribozyme can bind at least guanosine and thiomodified guanosine. We haven't tested other forms of polyphosphate, so it's unclear if it can use other polyphosphates besides trimetaphosphate.
Well, thank you so much, everyone. And I see that James just put in the link to the Slack, so we can continue conversation there. And we will see you all again in 3 weeks. Thank you again.